Hello everyone, this is part 2 of chapter 15, and we're talking about some of the cognitive pieces of things. I am going to do a brief introduction about some of these topics, and then I'm going to show you a few more things that were recorded by Dr. Levins. So where we're going to pick up today is with the idea of cognitive neuroscience, and I'm not going to do a ton here because again, I just want to briefly introduce some of these ideas, and then again, if you like this information, you can definitely go on and take a more advanced cognitive course. Again, I would take PSYC 3116. But the basic idea of cognitive neuroscience is sort of the interaction between, again, how we think, how we process information, how we make decisions, how we form memories, and how that intersects with the idea of what's happening biologically in our body and in our brain. And what's really neat, and what the next couple slides are about, is we have some really cool technologies that we've been developing in the last 5 to 10 years, and that are still coming out, that have really allowed us to get even better pictures than we used to. Because it used to be that some of the stuff we could kind of hypothesize about, and we really weren't able to confirm it until after the person had died, and we could sort of do a post-mortem autopsy and say, yep, it looked like that was what was going on. And see, now we can do some things where while the person is alive, we can do some imaging techniques and things that we just weren't able to do before. So for example, we can do some of the neuropsychological testing that we've been doing for a while, where we take someone who's had an injury and we kind of say, okay, what happens now that you've had this injury? So if you remember back to the previous lecture about like Phineas Gage, who had the frontal lobe injury, he had that big rod go through his frontal lobe. That's the kind of thing where we're like, okay, well, we know where you got injured, so now what kind of symptoms are you displaying? But again, that was about all we were able to do, is we just kind of had to wait until somebody had sort of a freak accident and then say, okay, now let's kind of study what's going on here. Now what we can do is we can take somebody that may or may not even have an injury, and we can look at what they're doing under just normal situations and find out what's going on. So we can look at connections during the brain while somebody's working on a particular task, for example, or while somebody's thinking about something or learning or whatever it is they might be doing. So we have different ways of imaging the brain that way. Again, you can do this while somebody is alive. You can take measurements of both one part of the brain. You can look at measurements during the entire part of the brain. You can correlate what's going on in a specific connection between different pathways of the brain that we know talk to each other, or you can do it between different brain regions to see what's happening. So there's a lot of ways that we now have that we just didn't have before to look at what's happening with somebody that's going on. So again, I just wanted to quickly mention that we have a lot better tools than we used to have. As the technology is developing, so is our ability to kind of understand what's happening in the brain and what's going on. So that's led us to some really other cool areas of being able to study. A couple others that I'm going to mention would be the idea of social neuroscience. So this is another blending between the idea of understanding what's happening in the brain, what's happening with your biology, but then understanding how that impacts how you interact socially with other individuals. And this goes back to the idea of theory of mind, where basically we are really good and our bodies are hardwired, thanks to a whole bunch of evolutionary processes, to try to understand what others are feeling. So I know that Dr. Levin spoke in the previous lecture about the mirror neurons. Our mirror neurons are quickly activated when we see somebody experiencing something, right? And hopefully that is in place so that we accurately try to understand, oh, they just experienced something happy, or oh, they just experienced something sad or scary or painful. And so it allows us to sort of quickly understand what somebody else might be going through, and that helps us understand what's happening into the world. It also helps us observe others' behaviors as well as listening to their words. And we can get a sense of not just what somebody is thinking and feeling, but we can also quickly go, wait a minute, what they're saying isn't matching their behavior. And we can sort of make judgments of, are they being truthful with me? Because understanding if somebody's being truthful with you is an important part of sort of this understood contract that we make with each other socially as humans. It also allows for us to have empathy with each other. And we know that empathy is correlated with the dorsolateral prefrontal cortex. The people that have injury to the dorsolateral prefrontal cortex legitimately cannot experience this concept of empathy with others. And it's also important for self-regulation. We use our emotions and we also try our best to control them in certain circumstances so that we don't just let our impulses guide our behavior. We sort of take a look at, okay, here's what I'm feeling, here's what I want to do, but let me just take a quick second and say, is that the best decision in this moment? Because I am able to use my frontal lobe to have made long-term goals, right? So yeah, even though that piece of cake looks really good right now, my long-term goal is to get my cholesterol level down. And to get my cholesterol level down, the doctor told me I needed to lose 15 pounds. So yes, that cake looks good in the moment, but it's not good for my long-term goal. So I can control my emotions and my impulses, again, by kind of stepping back, using some of that frontal lobe area that we talked about, particularly that prefrontal region, and actually use that for self-regulation. I'm going to show you a little bit of a video clip in just a minute. I'm not going to show you all of it. I'll let you actually go and watch as much of it as you want, but here's the link for it here. But we also know that this is why it's very difficult as people are growing and developing, because their prefrontal cortex is one of the last regions to fully come on board. Now, a lot of people will say it's the last to develop, and I really hate that word because it's not quite the accurate word. It's there and it's developed, it's just not connected. So as we talked about way early in the semester, and as you're going to hear about if you ever go take developmental psychology, especially if you take child or adolescent, what you're going to find is that most of the brain regions are sort of formed by the time that you're born. Right? We talked about the process, right? The neurons have to be formed, and they have to migrate to where they're going to go, and then they have to make connections, and then the last step is that they get myelinated after you've pruned away all of the unnecessary neurons and connections that weren't necessary. Well, the last brain region that gets that done, that pruning away of connections that aren't needed, that myelination to strengthen the connections that are needed, the last place that has that happen is the prefrontal cortex. And by last place, I mean that you're probably well into your late teens, sometimes even early 20s before that happens. So if you think about it, this is absolutely why little kids have a real hard time self-regulating their behavior. And this is why it's really important that as parents, we help teach our children how to do that, right? We start teaching them how to get that frontal lobe to start learning that kind of behavior. And then we don't just expect children to be able to do that. They biologically cannot do that on their own. It's not that you've done something wrong as a parent if your child doesn't do it. They legitimately biologically can't yet, right? Same thing with teenagers. Now, they should be getting a little better at it because they've gotten a little bit more myelination, but they're still not going to be 100% yet. So teenagers still do need a little more guidance than, say, a 25-year-old would need or a 35-year-old would need. What we also know is that understanding of ourself and our social interactions are linked together as a single mental interaction. This is a very important step as we sort of develop, and you'll see that some of the developmental models, if you ever go take developmental psychology, understood that without necessarily understanding that at some point, it's not just about me anymore. It's about me as part of a social world, and that becomes part of my self-concept. And that expressing my attitudes towards other ideas or groups reveals activation in the prefrontal cortex, as well as a few other places like the anterior cingulate and lateral parietal regions. So I'll show you this video in just a second about somebody who's had prefrontal damage. But one of the biggest issues that they have is they have trouble regulating their choices and their behaviors. And that led to a whole other field called neuroeconomics. And neuroeconomics is sort of a blend of a lot of different fields, including psychology and neuroscience, as well as the actual idea of economics. And it helps us to explain how we make decisions and how our brain sort of instinctively studies patterns of behavior in order to help us make decisions. So you don't even know that you're doing it, but on a very regular basis, you are constantly checking things out in the world, and you're constantly trying to make decisions based on patterns of things that have happened in your life. We have two systems for doing this. We have a reflective system and a reflexive system. The reflective system is very slow. It's very rule driven. You've done your best to try to keep yourself emotionally neutral while you're making the decision. So this is when you kind of sit back and you say, okay, what are the pros and cons of each decision? I'm going to pick the one that has the most advantages and the one that has the least disadvantages, versus the reflexive system, which is a much more fast and automatic, usually a very emotionally based choice that you're going to make. Again, when you do this, you're using different brain regions. So again, if you do make that more reflexive, fast, automatic decision, you're going to be using your ventromedial prefrontal cortex. If you do allow yourself to kind of take a step back and reflect on your choice before you decide, you're going to use more of the lateral prefrontal cortex, the medial temporal, and the posterior parietal cortex. So just a different series of brain regions that's involved to be able to do this. So again, what you're going to find is very interesting here, and again, going back to the video that I'm about to show you of the person that's had some damage, is that some of these brain regions that allow for this more deliberate, slow, rule-driven system get damaged in prefrontal damage, and so they don't have this reflective system, and so they only really can operate on this more reflexive system, and that creates a problem. All right, so I'm going to quickly show you the video clip, and then we will jump into a little bit more lecture done by Dr. Levins about some asymmetry in the brain and how the two halves of our main cerebral cortex don't function necessarily exactly the same. And we will go from there. So I will talk to you soon.
In 1861, one of the most celebrated patients in medical history died in San Francisco. His name was Phineas Gage, and 13 years earlier, he'd been the victim of a freak accident. He was buried without autopsy. But in 1994, using computerized brain imaging techniques, the husband and wife team of Hannah and Antonio Damasio performed a virtual autopsy using an electronic scalpel. Working with his railroad gang in Vermont, Gage was dynamiting large rocks when a tamping rod, more than a yard long and 1.25 inches in diameter, blasted up through his cheek, behind his eye, and out of the top of his skull. Astonishingly, he survived. He was still intelligent. His language and memory were intact. But he was a radically different person. As his colleagues put it, Gage was no longer Gage. The likable, hard-working, socially responsible character became untrustworthy, used profane language, and lied to his friends. It was as if the rod had destroyed the moral compass in his brain. The Damasios' detective work struck an immediate chord because several of their patients at the University of Iowa were, in a sense, modern Phineas Gages.
We have a large number of patients, neurological patients, who as a result of diseases have damage in certain sectors of the brain. It's a little hard to picture it, but you can think of them especially in the front part of the brain. And when there is damage in those areas, what happens is that patients become unable to read certain social clues out in society. So they will have difficulty picking up the significance of a facial expression or the significance of, for instance, a shrugging of the shoulders. And likewise, those patients will also not be able to perform in relation to what is expected in terms of the social contract.
You see those okay, Joe?
Meet Joe. Until a stroke in 1989, he was a high-energy, creative outdoorsman with a cheerful, fun-loving personality. Now, according to his wife, Dee, he's always in neutral. Intellectually, he understands what he's seeing, but emotionally, he doesn't connect. Like Phineas Gage, Joe has lost key components of his social brain. the evolved mechanisms that, behind the scenes, smoothly process and integrate reason and emotion have been damaged.
The goal of the game is to guess at which decks you can make the most money off of.
To test his ideas about the social brain, Damasio uses experiments designed with his colleague Antoine Béchara. They're called the gambling experiments.
In front of you there are 4 decks of cards.
You're hooked up to a polygraph machine and told to keep turning over cards from any of the 4 decks.
I'll give you some money each time you select a card.
With every card, you win play money. 2 of the decks pay $100, but they have an unexpected sting in the tail. You might have to pay the experimenter, sometimes as much as $1,250.
So you owe me $200.
With the other 2 decks, you win only $50, but the possible penalty is much smaller, less than $100 on average. The goal of the game is to make as much money as you can, so it has some of the same elements as the game of life. Uncertainty, some risk, punishment and reward, and a blend of reasoning and intuitions.
You win $50, but you lose $50, so it's even.
Initially, I did what most people do, favor the 2 high-paying decks. That is, until the experimenter started demanding big penalties.
So you owe me $50.
Having learned my lesson, I eventually curbed my gambling impulses and shuffled over to the safety of the other 2 low-payment, low-punishment decks.
You win $100.
Typically, over 100 plays, a normal subject will retreat to the safer decks about 70% of the time. Damasio's frontal lobe patients do the exact opposite. By halfway through the game, they're often bankrupt, which has happened to some of them in real life following their brain damage. Now, how can their judgment, their prediction of future consequences, be so disastrous? Damasio believes there may be a tantalizing clue in the polygraph responses. As the game went on, my graph showed a new trace that wasn't there at the beginning. I had the usual response after each penalty or reward, but I also began to develop a response just before picking a card from the risky decks. And that response increased as the game continued.
You win $50, but you lose $25.
In other words, according to Damasio, my brain was gradually learning to anticipate the penalty and sending an alarm signal, a gut feeling, if you like, that could be picked up by the polygraph.
The phrase gut feeling is actually a very good one. It just shows that people have been very wise for millennia in spotting the fact that some of their choices are very closely connected to emotions, and they're very closely following on emotions. In other words, the gut feeling is a marker for something that ultimately may be good or bad.
Now here's the key finding. None of Damasio's patients showed this anticipatory response. The neural networks that allow gut feelings about good or bad outcomes to guide rational decision-making are damaged. There's a break in the complex feedback loop of circuits that underpin reason and emotion. And so there's no way to make appropriate social judgments, and no way for the system to be calibrated or tuned by experience.
In this video, we're going to talk about the functional asymmetry that is present in the normal brain. One of the first ways that they started to identify functional asymmetry is through dichotic listening tasks. Dichotic listening is an experimental procedure for simultaneously presenting a different auditory input to each ear through stereophonic headphones. So you would present one type of auditory stimulus to the right ear and a different auditory stimulus to the left ear. What they found is that the right ear has an advantage for verbal information and the left ear has an advantage for musical information. You see a similar relationship for the visual system, that the right visual field advantage for language-related information and the left visual field advantage for nonverbal or spatial information. So if you recall, the brain is organized contralaterally. So language is processed on the left side of the brain, specifically your left temporal lobe. So if it's processed on the left, and the brain is organized contralaterally, then things that are heard in the right ear are going to be processed on that left side. So again, that aligns with that advantage for verbal information. Similarly, for vision, the right visual field is going to be processed on the left side. So then that would, again, align with this language-related advantage for information presented on the right side of the brain. And here, this is all shown. We have the right visual field, which is actually processed here on the left, and you have the left visual field that's processed on the right. So what we found from these early experiments is that, again, there's this language advantage for the left side of the brain, and there is a spatial, facial advantage for the right side of the brain. And in terms of auditory information, this is musical. And so, again, this left visual field, right side of the brain. So, and I said this is a spatial, facial, and that refers to an advantage for processing faces and spatial information and musical information. One of the ways that they further examine this functional asymmetry is by using split brain patients. Split brain is a surgical disconnection of the two hemispheres in which the corpus callosum is cut. We've discussed the impact of frontal lobe lobotomies, which of course are very, very negative. Well, when you cut the corpus callosum, that does not have the same debilitating effect. What you do is you prevent the two halves of the brain from communicating with each other, but the cognitive capacities of those two sides of the brain remain intact, and they find other ways to communicate with each other. So people that have split brain, or they have their corpus callosum cut, can still function quite well. What they find, though, is that language abilities, differentiate based upon where information is presented. So objects presented in the right visual field, that information goes to the left hemisphere. When that occurs, split brain patients can easily name that information. However, objects presented in the left visual field, this information goes to the right hemisphere, and because it goes to the right hemisphere, and that is not the language processing side of the brain, then split brain patients generally cannot name those objects. They can, however, make correct nonverbal responses. And you can learn more about that here, and as well as how the two hemispheres act independently. So when each hemisphere is shown a different object, in split brain individuals, the two hands will each select different objects as being the correct one. And you'll see some similar types of experiments in this very cool video.
We began our journey into the human brain here on the campus of Dartmouth College in New Hampshire. I'd come to meet one of the world's leading brain scientists, Mike Gazzaniga, and a man he's worked with for over a decade. A man with two brains. You've been working a long time with Dr. Gazzaniga?
14 or 15 years. It doesn't seem like that long, does it?
The collaboration began when Joe had surgery. And you had this procedure to correct an epileptic problem,
yes.
Is that right?
Trying to stop the seizures. I was having seizures like every day or so that's 2 or 3 a day.
To control Joe's epileptic seizures, a surgeon severed the connection between the two halves of his brain. Cutting the corpus callosum like this prevented the spread of the electric storms that caused his seizures. But it also prevented the left and right halves of his brain from communicating with each other. In the years since the operation, Joe's epilepsy has been under control. He now earns a living at an egg farm. And in his everyday life, he's largely unaffected by the fact that his left and his right brains work independently.
Do you feel any different when you think about something than you did any differently from the way you felt before the procedure?
It's just got a backup brain, that's all.
That's something everybody could use, right?
I found out how true that was right away when I was asked to draw a different shape with each hand. In a brain like mine, roughly speaking normal, at least all in one piece, the left half of my brain controls the right side of my body, while the right brain controls the left side.
Oh, no.
But because the two halves are connected,
Nothing wrong with that.
getting each hand to work independently isn't easy.
Well, we were saying that the fact that that Alan's hemispheres are connected, and that the motor messages from one are confusing the motor messages in the other.
I was just drawing an upside-down duck.
Okay.
But when Joe is given the same task, his two hands operate as if controlled by two separate brains. What's happening is that each half of Joe's brain is given a separate instruction. He's asked to fix his eyes on the cross in the center of the screen. Anything flashed to the right of the cross goes only to his left hemisphere. Things to the left go to his right hemisphere. Because the two don't communicate, each hand does only what its half of the brain sees.
Wow, look at that. It's really like two different people doing the same.
That's right. That's the idea.
Okay, Joe, I want you to keep your hands out in front
In an experiment that's now a classic in brain research, Mike Gazzaniga over 30 years ago used a similar setup to find out if the two halves of the brain are specialized to do different things.
Ship.
Joe is being flashed a word only to one half of his brain. Words flashed to the right
Storm.
are seen only by his left brain, and Joe can report seeing those words just fine.
Piano. Good.
But when a word is flashed to his right brain,
Didn't see that.
Okay. Joe, I'm going to ask you
but now watch what happens.
to draw that with your left hand.
I got me lost.
Why don't you try drawing another picture of it over here if that'll help you.
All full.
Oh, okay.
It's almost as though somebody has given him a secret communication.
That's
And
right.
now he knows that it's a telephone. Up until then, he was blind to it.
Exactly.
When Gazzaniga first did this experiment, it instantly proved that the ability to speak resides almost exclusively in the left hemisphere. Not until he sees what his right brain is drawing is Joe able to name it.
He said church,
After looking at the picture.
But he had to figure it out about as long as we did. That's really interesting.
So I think that's an awesome video for demonstrating functional asymmetry. And overall, it reveals that language is processed on the left and spatial abilities are processed on the right. Gazzaniga, who you saw in the video, is an expert in the role of the left hemisphere in language and thought. Unlike other animals, the speaking left hemisphere in humans acts as an interpreter. It is able to infer relationships among stimuli. And this also functions because language is fundamentally built to assign labels, to categorize, and this also underlies our ability to infer relationships. The language capacity of the left hemisphere gives a capacity for interpretation that the right hemisphere lacks. So it is through words that we're able to interpret and communicate what we interpret to other individuals. The right hemisphere, on the other hand, is highly involved in spatial processing, spatial reasoning, how we should move our body in space, attention for that information, and the processing of faces. So while the left and the right work quite obviously very intimately together, language labels and expresses the computations of our cognitive systems. And so we can then use these labels, these expressions to communicate with others and even to communicate with ourselves so that we can better regulate our actions. Humans are also a believing species, so the fact that we make inferences and have beliefs about sensory events is also likely to reside a little bit in the left frontal lobes because that is the primary region that has the foundational cognitive processes that are involved in labeling information, making inferences, and efficient and broad communication between various regions of the brain, the right and the left sides, sensory processing regions, is going to allow us to label information and make inferences quickly so that we can act adaptively in a range of situations. In this video, we're going to review some of the sex differences that exist in cognitive organization. You have already ideally watched the video that focuses on language lateralization, and now we're going to briefly talk about differences that can occur based upon sex or gender. Women tend to have larger volumes of dorsal prefrontal and associated paralimbic regions, whereas men have larger volumes of more ventral prefrontal cortex regions. So this would actually mean that the women are represented here in purple and the men are represented here in light pink. Women have increased cortical gray matter concentration in many regions of the cerebral cortex, and men gray matter concentration is more uniform across the cortex. Differences between white and gray matter between the sexes might reflect variations in cognitive organization. On average, females are better than males on short-term memory type tasks as well as verbal fluency type tasks. On the other hand, males on average are better than females on spatial relationship type tasks and on mental rotation type tasks. So why might this be so? One theory is that males might have better spatial skills because perhaps during evolution they had to tend larger territories than females so that they could hunt and bring back resources. On the other hand, females might have better language skills because during evolution, females develop good language skills to engage in social interaction. In the rearing of children, social interaction is highly, highly important. And early on, like many other species, it may have been that the women spent significantly more time with offspring than men. And this would encourage greater social interaction among women in comparison to men in the development of language skills. Females may also have been selected for fine motor skills, such as making clothes, which are related to language. Fine motor skills are also involved in the gathering component of early humanoid existence. The neural basis of these sex differences is also linked to hormones. Gonadal hormones influence the structure of neurons in the rat prefrontal cortex. So in the medial frontal cortex, neurons in male rats have larger dendritic fields. In the orbital frontal cortex, neurons in female rats have larger dendritic fields. These differences, however, are not seen when the rats have had their gonads or ovaries removed. So this suggests that particular hormones might be influencing the development of dendritic fields in men versus women. If you recall, hormones are essential for the differentiating effects of males versus females. Females are actually default in terms of testosterone needs to be received or be delivered at a particular time, and if testosterone is delivered at that particular time, then the fetus will develop as a male. If no testosterone is delivered, then the fetus will develop as a female. So hormones influence whether a fetus will develop as male or whether a fetus will develop as female. Hormones also continue to have developmental effects as animals age. Hormones continue to have developmental effects, and as hormones influence certain aspects of development that affect behavior, gender differences in behavior can also give rise to gender differences in cognitive organization. So what's interesting is that the differences in the brain that have been attributed to gender and sexuality are likely to be changing. So as gendered stereotypes change and what is considered male versus female behavior and roles, then it's highly likely that the brain regions that have in the past differentiated males and females will change and develop as well. There are also handedness differences in cognitive organization. So most right-handed people have language localized in the left hemisphere, but the opposite is not actually true for left-handed people. About 70% of left-handed individuals also have language localized in their left hemisphere, whereas about 15% have language localized in their right hemisphere, and the remaining 15% have bilateral representation of language. Anomalous speech representation is a condition in which a person's speech zones are located in the right hemisphere or in both. And prior research has found that building upon the increased communication that would need to occur between the various halves of the brain for language, Whittleson and Goldsmith found that the cross-sectional area of the corpus callosum was 11% greater in left-handed or ambidextrous individuals than individuals who were right-handed. So this could be to a greater number of fibers or thicker fibers or more myelin that is required for the continual language communication that would need to occur. For Chapter 15, we'll be covering synesthesia, followed by intelligence, and then we'll talk a little bit about consciousness. Synesthesia is the ability to perceive a stimulus of one sense as the sensation of a different sense, such as when a sound actually produces the sensation of color. A subset of the population experience synesthesia. It's basically the mixing of senses. It tends to run in families. The most common form is colored hearing. So when you would hear something, it would again produce the sensation of color. And most pairings are in one direction only. So for example, in colored hearing, seeing a color does not elicit the sound, but hearing a sound makes you see the color. And here is a nice brief video on synesthesia that is quite cool.
Imagine a world in which you see numbers and letters as colored, even though they're printed in black, in which music or voices trigger a swirl of moving colored shapes, in which words and names fill your mouth with unusual flavors. Jail tastes like cold, hard bacon, while Derek tastes like earwax. Welcome to synesthesia, the neurological phenomenon that couples two or more senses in 4% of the population. A synesthete might not only hear my voice, but also see it, taste it, or feel it as a physical touch. Sharing the same root with anesthesia, meaning no sensation, synesthesia means joint sensation. Having one type, such as colored hearing, gives you a 50% chance of having a second, third, or fourth type. 1 in 90 among us experience graphemes, the written elements of language like letters, numerals, and punctuation marks, as saturated with color. Some even have gender or personality. For Gail, 3 is athletic and sporty. 9 is a vain, elitist girl. By contrast, the sound units of language, or phonemes, trigger synesthetic tastes. For James, college tastes like sausage, as does message and similar words with the "age" ending. Synesthesia is a trait, like having blue eyes, rather than a disorder, because there's nothing wrong. In fact, all the extra hooks endow synesthetes with superior memories. For example, a girl runs into someone she met long ago. Let's see, she had a green name. These are green. Deborah, Darby, Dorothy, Denise. Yes, her name is Denise. Once established in childhood, pairings remain fixed for life. Synesthetes inherit a biological propensity for hyperconnecting brain neurons, but then must be exposed to cultural artifacts such as calendars, food names, and alphabets. The amazing thing is that a single nucleotide change in the sequence of one's DNA alters perception. In this way, synesthesia provides a path to understanding subjective differences, how two people can see the same thing differently. Take Sean, who prefers blue-tasting foods such as milk, oranges, and spinach. The gene heightens normally occurring connections between the taste area in his frontal lobe and the color area further back. But suppose in someone else that the gene acted in non-sensory areas. You'd then have the ability to link seemingly unrelated things, which is the definition of metaphor, seeing the similar in the dissimilar. Not surprisingly, synesthesia is more common in artists who excel at making metaphors, like novelist Vladimir Nabokov, painter David Hockney, and composers Billy Joel and Lady Gaga. But why do the rest of us non-synesthetes understand metaphors like sharp cheese or sweet person? It so happens that sight, sound, and movement already map to one another so closely that even bad ventriloquists convince us that the dummy is talking. Movies likewise convince us that the sound is coming from the actors' mouths rather than surrounding speakers. So, inwardly, we're all synesthetes, outwardly unaware of the perceptual couplings happening all the time. Crosstalk in the brain is the rule, not the exception. And that sounds like a sweet deal to me.
That's a pretty cool video that covers how interconnected our senses are. And it's great to learn about at the end of this class or towards the end of this class, because we've spent a great deal of time learning about the various senses, how the information is processed, where it travels in through the brain, and how all of the various sensory information get linked together to form representations. So in synesthesia, you have almost hyper-connected connectivity between the senses. Synesthesia has been found to be genetic, related to increased connectivity in these multimodal areas of the frontal lobes that receive input from the sensory areas. So again, these multimodal association areas, such as the frontal lobe, the temporal lobe, or the parietal lobe, the frontal lobe is the multimodal sensory area to trump all other multimodal association areas in the brain. And so it's likely that genes will influence frontal lobe connectivity to create this enhanced connection between the senses that leads to synesthesia. And here I'm going to lead into intelligence. We spend a fair amount of time being a bit worried about our intelligence because we think that it reflects a great deal about our abilities. And now that you've reached the end of this class and you have an idea of how information is represented in the brain, then I think it would be quite clear that general intelligence would be really difficult to localize in the brain. Investigations imply that cerebral connectivity and high glia to neuron ratio may play an important role in intelligence. And just in the way that synesthesia is this enhanced connectivity, intelligence is likely to be reflected in enhanced connectivity. Remember that the more experiences that you have, the more connections that you make. And individuals who are highly intelligent have spent a lot of time making connections, and this would be reflected in enhanced connectivity throughout their brain. We spent a great deal of time talking about neurons, but neurons also depend upon glia to support them. Nutrients are derived from glia, structural support. So there's also some research that suggests that the glia to neuron ratio might also play a role in intelligence as well. In particular, in the inferior parietal cortex, this has been linked to be involved in mathematical reasoning. One of the things that I think it's really important to know about intelligence is that there are multiple forms of intelligence. This is something that we would all have lay experience with. We know that there are multiple ways that you can be smart and intelligent. Culturally, at present, linguistical and logical and mathematical reasoning tends to be associated with general intelligence tests. And so these are fostered in primary and secondary education, but there are multiple forms of intelligence, and in particular, interpersonal and interpersonal are also linked with high career success because it reflects an enhanced ability to communicate with people. So overall, these are just some of the different types of intelligence that exist. We have linguistic, musical, logical and mathematical, spatial, bodily and kinetic, and as I mentioned briefly already, intrapersonal and interpersonal. These last two have also been linked with the idea of EQ, which is emotional quotient, and that is that, again, the ability to understand your own emotions, other people's emotions, motivations, desires, thoughts, would be very, very important for effective communication, leadership, teamwork, all things that are highly, highly important and especially prized in the corporate world. So individuals that tend to be high in intrapersonal and interpersonal skills are also thought to have this higher EQ, and that is also linked with greater success and increased happiness. There are also two types of intelligence with regards to divergent and convergent intelligence. Convergent thinking is a form of thinking that searches for a single answer to a question, such as 2 + 2 = 4. So this is what is measured in traditional intelligence tests, and people with temporal and parietal lobe lesions tend to actually perform poorly on these types of tests. Again, the temporal and parietal lobe, these are also key areas that are association areas. And both the temporal and parietal lobe, those association areas, if you recall, each are part of various processing pathways for naming and identifying and interacting with objects. So information that is processed visually will travel dorsally to the parietal lobes and ventrally into the temporal lobes. And both of those are important for spatially navigating objects and identifying objects. Divergent thinking, on the other hand, is a form of thinking that searches for multiple solutions to a problem, such as how many different ways can you use a pen. People with frontal lobe lesions tend to perform poorly on these tests. Divergent thinking is linked with problem solving. Divergent thinking is also linked with critical thinking. When you think critically about a problem, then you are trying to analyze it from multiple perspectives. And again, this multiple perspectives links with multiple solutions. So convergent thinking and divergent thinking are really quite different, and they are different types of intelligence that are prized. Both of them are highly prized, but certain types of careers would require greater convergent thinking and other types of careers would require greater divergent thinking. So while you're going through school and you are enrolling in your classes, we are trying to give you opportunities to increase both of your convergent thinking and your divergent thinking skills. Lots of the exams that we have obviously test convergent thinking, but then some of the writing reflection assignments that we have or critical thinking assignments, writing intensive courses or some of the oral intensive courses, those are also going to be ones that are going to require you to flex your divergent thinking skills. And we're going to end the chapter talking about consciousness. Why is it that we're conscious? We think of consciousness as this higher level awareness that is unique to humans. So we're very much concerned about what it is that then makes someone or something conscious. When we talk about consciousness here in this chapter, we're not going to be talking about sort of the metaphysical aspect of consciousness. We're going to be really talking about what consciousness is from a basic biological level. In terms of the basic biological definition of consciousness, this is a level of responsiveness of the mind to impressions made by the senses. So this is really closer to the medical definition in the sense that you are conscious if you are, again, responding to your environment, but consciousness is sort of this idea of level of responsiveness of the mind. So highly conscious individuals are going to be highly aware of their continual interaction with the environment. And evolutionarily speaking, consciousness would provide an incredible adaptive advantage because if you have an awareness of how you are responding to the environment, then this would significantly enhance your ability to learn and then improve over time. And not just learn and improve over time automatically, but learn and improve over time deliberately and with intent. And this deliberateness and with intent over hundreds and thousands of years is what would give rise to a lot of the complex interpersonal interactions and overall existence that we have with regards to culture, language, and our highly social and mental world. So consciousness emerges from neural circuits rather than from individual neurons. Consciousness is really best represented as being more than the sum of its parts. The greater the degree of complexity in neural circuitry, the greater the degree of consciousness. So what is the basic neural basis of consciousness? There are conditions in which people can process information without being aware of that information. And we've talked about a couple of them in this course. Blind sight, which we haven't talked about, visual agnosia, which the chapters have touched on, visual neglect, which we've talked about, and amnesia, which we've also talked about. There are also conditions in which people have conscious awareness of events that are not actually present. This would be phantom limbs, and this would also be hallucinations. So how do we then put these two pieces together? So we have conditions in which people process information that they're not actually aware of. And then we also have conditions in which people have conscious awareness of events that are not actually present. These two ends of the spectrum suggest that consciousness is likely a product of all cortical areas and their connections and their cognitive operations. So the representation of a visual object is likely distributed over many parts of the brain, and parts of this neural circuit must produce awareness of the visual object. So then if you have disruptions in that circuit, then you're going to have disruptions in being aware of that visual object. Likewise, if this visual representation is distributed over many areas and this visual representation gets activated in one area, then that would extend to the other areas even if that visual information might not be physically present in the environment, as such would occur with hallucinations. So consciousness really is putting all of these pieces together, all of the intricate neural processing that we have, being aware of how we are processing information. And in being aware of how we're processing information, we can also change how we interact with the environment. And all of this is dependent upon the interconnectivity of the neurons in our brain. And this ends chapter 15.